In this video, I want to recap what we know about this idea of the sliding filament model of muscle. So just as a refresher, we have this concept that this individual unit, this sarcomere, one sarcomere, is made up of a few different pieces. One of them, one of the very important ones, is the actin fibers, the actin filaments, which are these here in blue. Another of which is the myosin. That's this stuff in the middle. And another of which is titin, which is these spring-like things on the end, which you can think about as keeping the myosin centered inside the sarcomere. And the idea is that when a muscle extends or contracts, what's happening is that the actin filaments, these parts, are sliding relative to the myosin, which does not change its length. So I have a sketched out version of a couple different configurations of a sarcomere, where we've got the actin here in the yellow, myosin in the red. And remember that what's going on with myosin is that it has these little heads, and these little heads can grab onto actin and then pull this way and this way. So that is why muscles are able to contract to shorten their length. And so if we keep that in mind as we look at these four different examples of the length of a sarcomere, so what's going on up here in number one? Well, the sarcomere length is very long, and as a result, a lot of the myosin heads are not overlapping with any actin, and so they cannot produce very much force. When the muscle shortens a little bit, now we've got more overlap, so there's more area here and here where these myosin heads overlap with actin. However, so down here for number 2, we should get more force. However, for numbers 3 and 4, we can start getting two issues that crop up. Number one, as the muscle gets shorter, the actin filaments can overlap on top of each other like this. So if you imagine there's maybe another myosin protein down here, where now it was originally going to be able to grab onto this one, but now it's blocked here. So this overlap causes some reduction in the force production capability. And then if the muscle gets really short, you actually get the myosin starting to bunch up at the end. So one way of conceptualizing how we would expect this behavior to manifest in terms of the force production capabilities of the muscle is as follows. If we plotted the sarcomere length and that's just this, or up here, this length. If we plotted that against the force production by the sarcomere when you activate it fully, maybe you do this experiment with frog muscles or rat muscles or rabbit muscles, and you just put a bunch of electricity in so you know the muscle's fully activated. What you would expect to see, and indeed what you do see from experimental data, is a plot something like this, where up here, or let's just say in this region, the sarcomere is very long. That's like up here when we're not getting very much force production. You could imagine what happens right here is that the actin and the myosin don't overlap at all. So you'd have actin out here, and maybe the myosin is like this, and there's no overlap whatsoever, so they cannot produce any force. Then, as there's more and more overlap, you get more and more force, and at some point there's some optimal amount of overlap. Maybe that's right here, where you get the maximum amount of force. And then as the sarcomere gets shorter and shorter, now you get this interference like this, and maybe down here you get this bunching up like this, And at some point, the muscle gets so short that you can't produce any force whatsoever. Maybe that's when all of the myosin heads are just bunched up here, doing nothing useful. So you get this very characteristic force-length relationship. And this is one of the big strengths of the sliding filament model, is that what you would expect it to do corresponds exactly to what kind of experimental, what we see in experimental data on the behavior of muscles. So myofibrils are just sarcomeres stacked on top of each other. So everything I just talked about, that's one of these little guys, and then you just stack those end to end, and that's a myofibril. And then you bunch up a lot of myofibrils together, and that's a myofiber, or sometimes we just say a muscle fiber. So I tried to include maybe a more artistically talented illustration of what an individual muscle fiber looks like. You've got individual myofibrils here, each of these tubes, and the idea is that maybe one slice of those is a sarcomere, which is this thing that we were just talking about. So an entire muscle fiber, an entire muscle, in fact, should behave just like a scaled up version of one individual sarcomere. In the additional content at the end of next week, we also saw how the activation of muscles, meaning the thing that causes these myosin heads to reach out, grab onto actin, pull back in that power stroke, and then cycle through again, is fundamentally the calcium that is released from this structure called the Sarcoplasmic Reticulum. Often we'll just kind of draw it as like a bag of calcium, but it's maybe useful to remember that in truth it is this mesh that's all over the muscle and just ready to dump calcium into individual sarcomeres whenever it's needed. And we also saw how that release of calcium is mediated by action potentials from the central nervous system. In the notes, I indicated that the specifics of how the action potential goes down to the motor neuron and the neurotransmitters and the axon terminal, blah, blah, blah. All that stuff is not super important for this class, given that there are entire courses on that subject in kinesiology and motor control. So what we are going to conceptualize all of this central nervous system stuff is, and we'll talk about this in future videos, essentially a throttle that just controls whether calcium is getting dumped out or pumped back in. So that's the important structural stuff from last week. And in the next video, we're going to talk about exactly how this throttling of calcium into force activation, how we can think about that in a useful way to understand performance and clinical applications.